Well, no doubt everybody out there is still reeling from the distressing digression we had in the last lecture, away from our usual dichotomy, short-term good news, long-term stress and bad news, and discovering that chronic activation of pain systems, in fact, don't occur. You run out of beta endorphin, that whole fact that we finished with last time. What we do now in this lecture is get back on wonderful, comfortable ground. We're dealing once again with this dichotomy of ours, short-term activation of the stress response, doing a good thing, in this case, the theme of this lecture, enhancing learning and memory, chronic stress, doing just the opposite. Now, as with a lot of the other lectures, what we first need to do here is go over the basic workings of the system we're going to talk about. How does memory work? And what we'll do is first look at memory as a behavioral phenomenon, then how memory works at the level of whole sections of the brain, how it works at the level of individual cells, how it works at the level of networks. Now, the first important point here in looking at memory as a behavioral phenomenon is memory is not monolithic. There's not just one type of memory. If you will, there's a whole taxonomy of different types of memory. Short versus long-term versus remote memory, other subcategories. There is one that's going to be quite important here, a dichotomy between what is called explicit or declarative memory and implicit memory. Explicit declarative memory. You know a fact, and you know that you know the fact. I have a birthday in April. I am a mammal. I have a doctor's appointment next Tuesday. You know the facts sufficiently so that you can actively, consciously behave with that knowledge. Explicit declarative memory. You can declare that piece of information. In contrast, you have the implicit memory. And there's a number of different types of it. One example is what would be called fear conditioning. A type of association, a type of memory that you learn without being conscious, a type of memory, if you will, that your body learns, that you learn implicitly, unconsciously. Let me give you an example of this. I was in the 1989 earthquake in San Francisco, and it was not fun, and it left a lasting impression and implicit memory that I often find, which is I might be sitting someplace and some large truck rumbles past wherever I'm in, whatever building, and the ground vibrates a little bit, and I stop in my tracks wondering, is this another earthquake? And what that is implicit fear conditioning, because what occurred during the earthquake was 15 seconds or so of the stimulus, 15 seconds that have taught my implicit pathways something indelible. Other types of implicit memory, one that becomes very, very important in all sorts of realms, memory, once again, where your body almost knows better than your head does, just a metaphor, but where it's the memory of how to do things, how to do procedures, how to carry things off, how to do things that are adept and dexterous where you don't have to think about it. And what procedural memory is about is learning to do something automatic. Now, procedural memory comes in all sorts of domains. Like, for example, learning some sort of motoric task, what you initially do is have to deal with it as an explicit declarative piece of information, which then, with practice, transitions over into implicit memory. For example, you were learning some new part of a piece of music on the piano, and there's this difficult trill, and every time you're coming up to it a few measures before, you have this explicit declarative thought, which is, here it comes, remember, tuck in my elbow, lead with my thumb underneath, some such thing, this very explicit process. And then, one day you're playing it, and you suddenly realize, I'm 3 measures past that trill, and I didn't have to think consciously about it. And that's the day that information got moved from an explicit pathway into an implicit one. That's the first time where, in effect, your hands know it better than your head does. So the process of learning some motoric pathway typically takes the route of moving from explicit information to implicit. Something that's interesting is the system is lousy going in the opposite direction, going from implicit knowledge, information, procedural information that you've got to the point your body knows it better than you do, and forcing it back into a declarative channel. And we can see this with a very, very simple experiment, one that you should not carry out alone, which is to walk down a flight of stairs. And right near the bottom, maybe 3 steps from the bottom, think in this declarative, explicit way. So what exactly am I doing when I'm going down a flight of stairs? Am I beginning to shift my weight over to my right knee at this point, or am I still holding something in my left butt? And what's going to happen is you're going to fall down those stairs, because we have not done stairs in an explicit, declarative way since we were about 3 years old. So often, the process of getting new automatic information, going from explicit into implicit pathways, forcing it in the opposite direction is real clunky and doesn't work very well. So now we can transition to the actual brain. What parts of the brain are involved? And take Intro Neuro 101 anytime in the last millennium, and you start off with a part of the brain called the hippocampus. Hippocampus is intensely involved in memory formation. Hippocampus, some Latin name hippocampus means seahorse, which shows you how pathetic neuroanatomists are, because they never get outside the lab, because hippocampus doesn't look like a seahorse at all. It actually looks like a jelly roll, but I guess nobody knows Latin for jelly roll. Hippocampus is this part of the brain that does an aspect of learning for you, of a certain type, of the declarative memory type. And you can see this specialization in a number of different settings. You, for example, can see patterns of brain activation. You put somebody in one of those scanners where you can see what part of the brain is being metabolically active, deal with a new fact, trying to learn it in a declarative way, and it's the hippocampus that activates. Look at something that's already in an implicit, your hands know it better than your head does, procedural way, it's a different part of the brain, the cerebellum, that typically activates. So the hippocampus is about declarative knowledge. You can also see this in a much sadder circumstance. You look at somebody with Alzheimer's disease. Alzheimer's disease predominantly damages parts of the cortex and the hippocampus, demolishes the hippocampus in lots of ways. And what you see with people with early-stage Alzheimer's, it's the declarative memory that's shot, the procedural is still good. And that's where you have somebody where he could not tell you what decade it is and where he is, but this retired plumber can still fix a sink trap without even thinking, because that's procedural. So hippocampus and the associated cortex specializing there in declarative memory. Now, this has also been shown in one of the most remarkable incidents and sort of remarkable chapters in all of neurobiology, the story of a man, the only person in all of history where scientists, where doctors intentionally took out his hippocampus. And this was a man who was only known to the literature as HM by his initials, and he is in every textbook. HM in the 1950s had a rare, horribly intractable form of epilepsy that was running amok through his hippocampus. The drugs available at the time weren't controlling it. It was clearly going to kill him, and the very desperate decision was made to go in surgically and remove his hippocampus. That was done, very famous neurosurgeon, and in the aftermath of it, the seizures predominantly went away. It officially worked, and in the aftermath of that, HM's ability to learn new declarative information was very, very much impaired, virtually entirely impaired. Everything he had learned before the surgery, everything filed away, was still intact. He remembered his childhood. He remembered the name of his mother, things of that sort. Everything was there because it was stored someplace other than the hippocampus. It was acquiring new information where things didn't work. There was, say, a 30-second memory span. There wasn't this transition from very fragile short-term memory into more permanent long-term memory because there wasn't a hippocampus. And this was absolutely remarkable to see. I actually met HM once, Of course, he never remembered me. I met HM once. It was when I was doing research in a laboratory at MIT, and he happened to be most intensively studied by some neuroscientists at MIT, and it was one of those like dream moments where you're sitting there in the lab and somebody runs in and says, guess who's out in the hallway? HM, oh my God, let's go meet HM. This is like taking Pavlov's dogs for a walk. So you run out in the hallway, and there's HM, who's this perfectly gregarious man, and he introduces himself, and you shake hands as saying, hi, my name is Henry, and that's great. And somebody slams a door down the hall, and you glance over, and you look back, and he says, oh, hi, I don't believe we've met. My name is Henry. And you could stand there doing that all day long, utter damage to his declarative memory processes. Now, it was HM that first got people attuned to the fact that there's more than one type of memory. There's this explicit, and there's also this procedural. HM could continue to learn procedural tasks. He could learn how to do things with his hands at least as well as any control subject. He simply didn't know he had learned it. So you would give him some puzzle task kind of thing, and because of his explicit memory problem, he had no memory he had done it, and he would say, well, I've never seen this before, but I'll try, I'll do my best. And then he whips through it incredibly fast because he has the procedural memory stored elsewhere. What about HM? In 2009, HM died in his 80s, and people who had been waiting to study his brain for decades moved in at that point. HM, and early in 2010, a virtual religious rite ran through the neuroscience community. The neuroanatomists and neuropathologists who were going to study his brain went through the first step, slicing his brain and mounting the slides to begin to examine the brain tissue microscopically. And in a remarkable event, all of this was broadcast live, streaming video online, where tens of thousands of scientists watched this. And there was something intensely moving about sitting there watching the brain of HM and the involuntary sacrifice of this man for science, watching the brain being sliced. So HM teaching us tons about the hippocampus. Shifting down at this point to the level of individual cells. What's going on with cells in memory? Well, people used to know just what was going on around 1900, which was this view, every time you make a new memory, you make a new brain cell. And that memory is contained in that brain cell, that neuron. That one fell by the wayside very quickly. Then there was a new view there. There are these connections between neurons, technical term synapse, don't worry about it. There's these connections, those cables that we've now seen, say, in the diagram in the last lecture. And the notion there was learn something new and you form a new connection. And that dominated for a while, except that went by the wayside because that didn't seem to be happening much. And what happened by the 1950s is what came to be the dominant view, a scientist named Donald Hebb. And when talking about memory, if you don't mention Hebb's name, they take away your neurobiology license, because he had the most wonderful dominating insight, the notion that a memory isn't about a new neuron or about a new connection. It's about strengthening pre-existing connections, making those connections more excitable, potentiating those connections, making them more excitable, and potentiating them so that they last long-term. And the jargon in the field winds up being LTP, long-term potentiation. So that's wound up being sort of the dominant model, and an enormous amount of work has gone into figuring out what that LTP, that long-term potentiation, is about. So some sense on the cellular level now. But now a level that's come to be really interesting to lots of researchers in the field, the notion of networks of neurons storing memory. Okay, so that Donald Hebb view, that memory is about you stimulate a pathway over and over, you learn something, and you then change the excitability of this synapse, and that's where the memory is. A single memory is not stored in a single connection between neurons. Instead, it's stored in networks. And the whole notion of these networks is information is diffusely distributed. Information, a memory, is not contained in any single neuron or any single connection. It's contained in patterns of activation. And what you see is a characteristic of memory which absolutely proves how it's contained in networks. You're sitting there in a party, and you're trying to remember the name of that person sitting across from you, and you can't remember their name, and what you begin to do is try to pull out that memory by association. How many syllables does that person's name have? I'm remembering it starting with a B sound. Let's see, where was I when I learned that person's name? Who is I with? You're tapping into associations. You're tapping into the people with this number of syllables in their name network, and people who have their name starting with B, and people who I met in this setting, and somewhere at the intersection of all those networks, that name will suddenly pop out. And that's what memory, that's what pulling out something is often about in terms of these networks. And what that begins to explain is something remarkable you could see with Alzheimer's disease, which is early on in the disease, it's not that memories are lost, it's harder to pull them out. It's harder to evoke them. And you can see this in all sorts of ways. You get somebody where, for example, you are trying to tap into their orientation to space and time. You have an Alzheimer's patient, and you will give them an orientation test. You will say, well, do you know what the date is? Do you know where you are? Do you know how many children you have? The name of your spouse? Some such thing. And then you will ask them a question, one which is pertinent right now when this is being taped, which is to ask them, oh, and do you know the name of the former president? Not there. Completely gone. They can't evoke it. And now what you do is prove that that information is not contained in a single neuron that died, that the information is in a network. You begin to tap into the intersection between that network and the other ones. You prime the person. You say, okay, let me help you out a little bit. The name was one syllable. Nothing. Doesn't come out. So then you give them a little stronger priming, tapping into yet another network, saying, okay, it's a one-syllable word, and it's something you might encounter in a park. Nope, doesn't come out. Then you give them the strongest type of priming. You actually force them to choose. You say, okay, so was it President Bench, President Tree, President Shrub, President Bush? Bush, that's right, the kid, the son, Bush, President Bush. And out it pops, showing this characteristic early on in Alzheimer's. It's not that the memory is lost. It has taken more work to pull it out of the networks because the networks have lost neurons, the networks have weakened. So that gives a sense about how memory works at the level of whole regions of the brain, the hippocampus, and it's not just the hippocampus, but hippocampus being very notable for this declarative memory. What's going on down at the cellular level? Memory does not consist of making new neurons, does not consist of making new connections, but strengthening pre-existing ones, and how those strengthened connections play out over the course of lots of neurons, networks of neurons, making those connections with each other. Ironic ending department, it turns out, that ancient, laughable notion that memory consists of making new neurons, or even memory consists of making new connections, that's ridiculous. That's medieval neuroscience. In the last few years, there's increasing evidence that that actually does occur some of the time. A huge revolution in neurobiology has occurred for the last, centuries. If you're learning basic neurobiology, one of the facts that gets hammered in immediately is the adult brain doesn't make new neurons. You're stuck with whatever number you got early on, and all that happens from there is you go downhill with fewer of them. This revolution being that it turns out the adult brain, even in humans, does make new neurons, adult neurogenesis. And remarkably, the place in the brain where it happens the most is in, you guessed it, the hippocampus. And at this stage, there's beginning to be evidence that certain types of memory acquisition require the formation of new neurons. So here we have both learning the lessons of history and having it repeat on you. Here we are back again, the notion that some of memory formation actually involves forming new neurons or forming new connections, as it turns out. Nonetheless, the bulk of what goes on appears to be that strengthening of pre-existing neurons. Okay, so we've got basic memoryology under our belt here at the systems level, networks, individual cells. So what happens with stress? What happens? You know exactly what happens, which is short-term, you have memory enhanced. Short-term, stress does great things for your learning and memory. And we saw that starting in the very first lecture. Short-term, what do? You increase the delivery of oxygen to the brain. You increase the delivery of glucose. Those are exactly the sorts of things that make for a perkier brain that is more able to do the very energetically costly process of all that excitation business. What's that do to? That's the increased heart rate, blood pressure. You dilate, you loosen up some of the blood vessels in critical areas of the brain, deliver the oxygen, glucose up there, and suddenly you got a brain that works better. What else? With the onset of stress, you suddenly have those neurons that undergo that long-term potentiation business, that business about these connections becoming more excitable in the hippocampus, and suddenly LTP happens more readily. And this could then be shown on the level of actual learning and memory, the sort of thing where you look at, does a stressor make it easier to remember certain things? And this was shown in a magnificently elegant study carried out by two scientists, Larry Cahill and James McGaugh, University of California, Irvine. McGaugh, one of the senior figures in the field. And here's the study they did. Totally elegant. You take a bunch of control volunteers, and what you do is you tell them this 12-sentence story. Boy and his mother go walking through town. They pass the grocery store. They pass the hardware store. They cross the street. They go into the hospital where the mother works there. The mother shows the boy how the x-ray machine works, and on and on. The first 4 sentences being, they cross the street up to that point, then in the hospital. So that's the first version. The second version, the experimental subjects, what happens with them is one part of the story is different. A boy and his mother are walking through town. They pass the grocery store. They pass the hardware store. They begin to cross the street when the boy is struck by a huge truck, and the boy is rushed to the hospital where they rush him to the x-ray room and x-ray him in this big... You'll notice the first 4 sentences are the same, and then it goes into a much more emotionally arousing, a much more stressful story. And what Cahill and McGaugh showed was come back a couple of weeks later and see what people remember about the story. The people who got the first boring version, the mother goes and shows the kid the x-ray machine, they don't remember it very well. You get the people who got the much more stressful, emotionally arousing version, and they remember it better. But the part they remember better is starting when the child is hit by the truck. That's the arousing, stressful part. That's the part that gets filed away as suddenly the oxygen and the glucose is coursing up into the brain. So what causes this to happen? This has some responsiveness, some involvement of glucocorticoids, some involvement of the sympathetic nervous system predominantly, and in the final elegant touch to the study, they showed exactly how the sympathetic nervous system plays that role, which is they gave these volunteer experimental subjects a little bit of a drug which blocks activation of the sympathetic nervous system. A drug which blocks a certain type of receptor for epinephrine, adrenaline, and this is a drug lots of people have heard of and use. It's called a beta blocker. Beta blockers tone down sympathetic tone, drugs like propranolol. And what they did at this point is they either had people get a placebo and hear the horrifying, really distressing, stressful version of the story, or people who were given a beta blocker and heard the same story. In other words, they went into hearing the really upsetting part of the story with their sympathetic nervous system damped down. And those folks didn't have preferential memory of that event. What that's telling you is this short-term enhancement of memory driven by the sympathetic nervous system is, number one, specific to the more arousing parts of the memory, and as just shown with that experiment, yes, indeed, involves the sympathetic nervous system. So we've got our short-term response to stress, a 30-second story. Now we switch over to our more chronic scenario of stress. More chronic, and what you begin to see, as we would absolutely guess, is a more sustained stressor disrupts memory. A more sustained stressor, how would you show that in lab rats? They have to learn some sort of maze, that kind of thing. How do you show it in humans? All sorts of experimental settings. You force somebody to go through a stressor, like giving a public speech, and then afterward you see how well they remember something from beforehand. And that might be viewed as stressful in this case, but fortunately I'm a computer-generated image. But for most people, doing something like this, you would begin to disrupt memory because it's stressful. What do you see? Once stress is going on for a few hours or so, no longer are you delivering more glucose and oxygen to the brain, you're delivering less. No longer are you enhancing LTP, that long-term potentiation, you're disrupting LTP. And exactly where you don't want to have it disrupted, in the hippocampus. Next, you actually, with more sustained stress, begin to do something remarkable, which is you change the structure of neurons in the hippocampus. You cause them to atrophy. You cause them to atrophy, those long processes. We've been hearing all about those long processes with which a neuron talks to another neuron. What are you doing there? You're disconnecting circuits. So suddenly, what circuits there are not very excitable, disrupting that long-term potentiation, and you're making for fewer connections because you're disconnecting, you're atrophying these neurons. But oh, that's not enough. More things go wrong. Back to that revolution in neurobiology, back to that finding that the adult brain, the adult hippocampus, can make new neurons. When does that occur? In response to all the logical things. Learning something new, environmental stimulation, exercise, very interestingly, estrogen as well, a whole bunch of things that stimulate neurogenesis. And the two surest ways to shut down neurogenesis, lots of glucocorticoids and lots of stress. So that's another domain in which you pay a price. Then, moving on to a next realm, what you have with sustained stress, and glucocorticoids play this role, is you endanger neurons in the hippocampus. You take hippocampal neurons and you make them a little fragile and a little bit wimpy and a little bit nauseous and green around the gills. What you do is you take a hippocampal neuron and you put it right on the edge of a cliff during sustained stress. At that point, if nothing else happens, the neuron recovers. Everyone lives happily ever after. If at that point, though, you push the neuron with something else, it's that much more likely to go over the edge.